like a question I can answer. But no, I think it's two questions. Like, bro,
All right, folks, good afternoon. The week is going well. So the exam viewing is ongoing. Please do set aside time to visit Logan or John's office hours so you can view exam 3. Address any questions you had, questions about content, questions about the grading, and questions about how to approach these types of questions. It's still not too late. I've got one more exam, a major component of the fourth grade. If you have questions about how to study, how to use your time, how to approach the questions, Brianna and Logan are ready for you. All right? So please stop by. All right? Tomorrow, rather next week, we continue and do that process. So if you haven't noticed already, the chapters are going really quick in this section of this quarter of the course. And what that means is we're just covering, well, I think number one thing it means is the chapters are shorter. These are shorter chapters in this section. And the other thing it means is we're covering not all the content in these chapters, right? So please don't freak out about the number of chapters. I hear that from students at this time in the course, like, oh my God, we're covering so many chapters. Exam 4 is over so many chapters. No, I mean, numerically, yeah, maybe it's several chapters, but in terms of lectures, and what are you responsible for in this course? What we covered in lecture, no more and no less, as we said from the beginning, all right? So keep the perspective correct, I think, and then it helps, so you don't freak out. So that said, we are completing cell division today. Right? We actually started chapter 13 on Monday. We're going to conclude that today, and then we start chapter 14. So classical genetics, which is our next topic, is also covered over 2 chapters, just like cell division is covered over 2 chapters. Right? So that's why I refer to 12, 13, 14, 15 like that, because it's really one topic. All right? So we get halfway through chapter 14 or so, and then we'll pick up there on Monday, and then we get just come to the end, I think, of chapter 14, and Wednesday we'll go on to 15. And then go on to 15, and then go on to 16 on Monday, I think it is. Is that right? Or Wednesday next week, okay? You get the idea. So several of these lectures we're covering 2 chapters, finishing one, beginning another, and just kind of keep up with it, just roll with it. All right? That's all I have, folks. As usual, stay on top of your lecture content, right? The notes you're taking in class. Work the review sheet. Do it now. Don't save it till the end. Use the textbook as your need dictates, as your time allows. And then finally, reminders about assignments. You have mastery number 7, due midnight. You have quiz number 7, due Saturday, and also the last intervention, also due Saturday. Okay, we're done with the intervention. Not done with the study. We got to do the post surveys to complete that, but anyway, no more intervention. Okay, group work is okay. Question about anything before we get started? No? Okay. If not, we do have a full lecture. Let's go ahead and get going. So, in our last lecture, in order to begin talking about meiosis, right, the other type of eukaryotic cell division, we laid some groundwork, right? We talked about chromosomes again, and then we turned to human chromosomes as an example. Give us something to look at, give us something to count, and so forth, right? And it was here that we introduced this concept of homologous chromosomes. Okay, so this is super important, folks, in order to understand the process of meiosis, and this is a challenging topic. I think students have trouble keeping meiosis separate, really understanding what meiosis is about. This is a classically challenging topic. This is really important, right? So if you look at this and you say, oh yes, human karyotype, 46 chromosomes, 22 pairs, and you kind of, it makes sense to you, then you're good. I think you're where you need to be. You look at this and you go, oh, it's that figure you covered, oh, I'm not sure why is it every, blah. Then we should talk about it. Okay? If it's scaring you, you're not sure what this is, let's talk about it. Let's talk about it in office hours, talk about it in TA office hours, whatever. Get those questions answered. All right? Before we close it out, there are some terminologies that are important that we need as well before we go on to meiosis. So, somatic cells are what again? Body cells, right? That literally means body, right? Soma means body. And so, yeah, these are just body cells. They're like most of the cells of your body, right? And most of the cells of your body indeed do have two sets of chromosomes. What are we talking about with sets? What is a set? Where does a set come from? From each parent, right. You have two parents, right? Again, sexually reproducing organisms, we are. So each parent is going to give us a set. We have two sets. Okay, it's very important you understand this term set. And a few years ago, not to find a spin on it, just to be clear, right? If, for example, every first chromosome of each pair came from mom, well then that would be a set, right? So like all of those chromosomes could be one set. Now I think that's not the case, right? I mean, I don't think the microscopist who created this karyotype had any idea, which is moms and which is dads, right? Which is moms, which is dads. And so it's a little bit of a sleight of hand here, right? But perhaps if those were all from mom, then that would be one set, right? And the other chromosome not boxed in would be the other set. Does this make sense? Okay, again, super important you understand what we're talking about. And then the figure's going away. So any questions? Anything? All right, if not, then of course the sets are from the two parents, so one maternal, one paternal set. And that's why we have pairs, right? We have two sets, and so we have pairs. The homologous pairs from that. Now, if lowercase n were to represent the number of chromosomes in one set, then somatic cells are what? 2n was what I think someone said it. Right, so we have two sets, we said, and so if n is the number of chromosomes in one set, then 2 times that number would be the number of chromosomes we have, right? The somatic cells contain. Okay? So our somatic cells are 2n then, by that reckoning. And we have a name for that. 2 sets, 2n, we call diploid. We call that number of chromosomes diploid, the diploid number. Okay? Now that is in contrast to what gametes end up with. Remember, gametes are, the egg and sperm, or the ova and the sperm, right? Are the gametes, female and male, respectively. They're the products of meiosis. And the products of morphological changes that occur to produce an egg and to produce a sperm. But meiosis is a big part of making an egg and a sperm. And what meiosis does is ensure that gametes only have one set. They have n number of chromosomes, right? Exactly n, not 2n, 3n, 5n, just 1n or n, right? And we call that condition what? Haploid, right? That's the haploid number gametes have one set, or they're haploid. Gametes are haploid. Okay? That makes sense? So what is N in humans anyway? Okay, so it's the number of chromosomes in one set. But for humans now, specifically, what is N?
23.
23. Okay, you gotta be like that with that. And for humans, 23, of course, yeah. And that includes 22 autosomes and one sex chromosome. Right? Each parent gives us 22 autosomes and one sex chromosome. Right? And I don't know, we mentioned this, but so meiosis, we said, is going to separate homologous pairs. It also separates our sex chromosomes. So sex chromosomes are not a homologous pair, right? They're not homologous. But they are a pair in the sense that they get separated and then the gametes end up with one or the other. Okay? So again, the 23 includes 22 autosomes, one sex chromosome. 2N, of course, 46, and then that would include 44 autosomes and the 2 sex chromosomes. So 46. Okay. Any questions about these terms? How they relate? Super important to make it part of your vernacular. All right, so we'll end this introduction on this figure because this lovely life cycle shows how these terms kind of play into our, our life cycle, us. Okay? And your textbook includes, I think, some other life cycles of other species. Bio 2, please. Let's focus now on, what is most relevant for us. Okay, and literally us in this case. All right, so we are diploid. We are multicellular to be sure, and we are diploid. That means that most of our cells have a 2N number of chromosomes, which is 46. But in these very special places, in ovaries, in female, in testes, in male, meiosis happens and produces cells that are what? Haploid, right. Right, that's what meiosis does, right? So it results in gametes that are haploid, right? In other words, they have N number of chromosomes, 23. That's true for the egg, and that's true for the sperm. Of course, that's temporary situation. And in fact, an egg will get together with the sperm. Sexual intercourse, of course, happens, and then an egg and sperm are united. Fertilization, that's called, ensures that the chromosome number is restored to 46. Okay? Or the diploid number 2N. All right? Now, what happens then? Oh, and what is a fertilized egg called? Zygote. Yeah, it's a good term to know, zygote. Basic stuff. Now, what happens then is not meiosis, but what? Mitosis and mitosis and more mitosis to produce a multicellular individual, right? And that takes 9 months, and then here's an individual now, and then that process continues, even more mitosis through early stage of growth, and then adolescence, and then culminates, I guess, with adulthood, right? At which point the mitosis comes down. All right, so mitosis and development, differentiation of the cell types, right? We get many different cells. From one cell type, we get many different cell types, ultimately produces a diploid individual. And I adjusted that number, I think 200 trillion on my slide, a little bit high. So I think on an average, about 30 trillion cells is how many, only 30 trillion cells is how many cells we have on average. Again, it's a little higher or lower depending. Okay, any questions about human life cycle? Again, the only one I think you need to know. And how these terms, again, apply. All right, if not, then let's now finally go on to talk about meiosis. Right? Armed with this knowledge and terminology, let's do it. So, meiosis, unlike mitosis, involves two consecutive rounds of cell division. Okay, note the wording there. Two consecutive rounds of cell division. That's not two cell divisions. In fact, it's not two cell divisions, it's a different number of cell divisions if you do the math. But it's two consecutive rounds. And these are simply called meiosis 1 and meiosis 2. Roman numeral 1 and 2. So, over the course of that process, we will reduce the chromosome number by 1/2. Again, that's the goal of meiosis. And how is that accomplished? Again, by separating the, what do we separate? Chromosomes. We need more though. We need more than that. Separate the homologous pairs. Right. Again, use that wording now that you know what that means. Okay? Yeah. By separating the homologous pairs, meiosis does that. Okay? All right. Let's get into it. Now, meiosis is a type of cell division at the end of the day. Therefore, it is preceded by what cellular process? DNA replication. Nicely done. Okay. So I call that step 0 because that's not part of meiosis. Remember, that happens in, okay, I'm done with this. And that happens in the S phase. S as in Sam. S phase of the cell cycle. Right? So be sure you've gone back and completed the video lectures because that's when we talk about the cell cycle. But indeed, that is when DNA replication occurs. All right, then let's fast forward. We got G2 and then we go into an M phase. But M at this point is not mitosis this time. M is for meiosis. The steps of meiosis are as follows. Step 1, meiosis 1 occurs and separates the homologous chromosomes. Well, look at that. Out the gate, we separate the homologous pairs. All right. And what chromosome number do we have now? We were diploid, right? We were diploid, and then now what are we? Haploid. That's correct. We've already gone from diploid to haploid by separating the homologous pairs. Yes. And this really begs the question, why? Why is there even more to meiosis if we've now created haploid cells? That's why, yeah. Because every cell division is preceded by DNA replication. Does that include meiosis? Another type of cell division. Yes, it is. So, you gotta deal with that. You gotta deal with the positive DNA replication, which is, of course, sister chromatids, right? So we still have sister chromatids to deal with, and that's what meiosis 2 does, is separate the sister chromatids, okay? Mopping up meiosis, if you will. Make no mistake, the business end of meiosis is what? Absolutely meiosis 1. It separates the homologous pairs. It takes us from diploid to haploid. The only reason there's more is what?
Sister chromatids.
Because we have replicated DNA. Because we have sister chromatids, we gotta separate them. Okay. Question? All right, meiosis in a nutshell. And there's a figure which summarizes this. Again, painfully simple and really the simplest possible sexually reproducing organism here would have 2 chromosomes. Right? You gotta have a pair, right? You have to have an even number of chromosomes in order to do meiosis. That's an expectation. And this is the simplest possible even number, right? Okay, so before anything happens, right, we have that homologous pair. Now, before meiosis begins, what would occur? Everybody, DNA replication, that's included on the figure. There it is. DNA replication produces replicated chromosomes. Ah, there they are. So that when meiosis 1 occurs, it does take us from 2 chromosomes to 1. It does take us from diploid to haploid, but we still have replicated chromosomes, which must be dealt with in a separate round of cell division. Okay? Meiosis 2 separates sister chromatids and then gives us this, right? Gives us 4 daughter cells, at least in theory, right? There it is. Each of which has half the number of chromosomes as the original parental cell. Parental cell had 2, these have 1 each. Okay. How many cell divisions did we just see? 2 rounds. How many cell divisions actually? How many do you see?
4.
How many times did one cell divide into 2? Twice? Careful. How many cell divisions? Oh. Okay, it's 3. Right, I've heard it, we gotta go. All right, one cell divides into 2. Cell division. Another cell divides into 2. Another cell division. And a third, that's 3. Folks, don't overcomplicate. I know you want to say 2. I got it. Because meiosis 1, meiosis 2, meiosis 1, meiosis 2. The question is, how many cell divisions did you just see? There it is. Easy? Sure it is. Okay, very good. All right, big concept. Let's just frame it as such. Meiosis 1 is going to separate the homologous chromosomes. How it does that is worth some consideration. Okay, this is unique now. This is not something we saw in mitosis. This is not something we see anywhere else, in fact, and so here it is. Early in meiosis 1, and understand, both meiosis 1 and meiosis 2 have all of the parts you've learned for mitosis. And you got a prophase, you got a prometaphase, a metaphase, an anaphase, a telophase. If you're good with that, I mean, do you really now have to memorize, 5 more such phases for meiosis 1, another 5 for meiosis 2? It's the same deal. It's the same stuff happening again, but with a very specific goal. Okay, so in case in point, in prophase 1, chromosomes condense, to be sure, as in mitosis, but the homologous chromosomes pair with one another. That's different. That's unique. That's not something we've seen before. Moving along now to metaphase 1. Remember, metaphase chromosomes are where? In the middle. Meta means middle, right? But pairs now. Pairs will align in the middle. And in anaphase 1, the pairs separate. Okay. So it's all about what the pairs do is meiosis 1. Okay? And let's take a look. Let's take a closer look and see how that happens. So in your text, figure 8, 13.8, right, is going to be the big splash page, meiosis, every single, prophase 1, prophase 1, prometaphase 1, metaphase 1, anaphase, everything is there. And I leave it to you. Okay, I'll leave it to you to enjoy, to kind of convince yourself, okay, this looks a lot like mitosis, I can see how that's different, this is different. Let's go to figure 10, which I think gets the point across and also compares and contrasts with mitosis. Okay? So leave 8 to you, which I think you're more than capable of considering. All right, this is that figure, and it tells us that this is a parental cell. What that means is it's a cell before cell division occurs. Before any cell division happens. All right, and clearly not, is it a human cell? Excuse me, is it a human cell? What do you think? No, I don't think so. It's not enough chromosomes. But it's more than that 2-chromosome cell, right? And how many chromosomes does it have? That's 6. Yeah, I see 6 in there. I hope you do as well. So what could we say about the diploid number of this unspecified species? What is it? Okay, so remember, the diploid number simply means the number of chromosomes in a diploid cell. What would that be? It would be 6. Yeah, again, this is before cell division has occurred. That would presumably be a diploid cell, and so it is 2n number of chromosomes, which means 6 in this case. Okay? So look, there's 2 ways you could answer that. First, what is the diploid number? Well, it says parental cell. No cell division has occurred. That's one way. The other way is you could look at the coloring they did for the chromosomes. Okay, what are the colors of the chromosomes here? Blue, red, yeah. Okay, so what I think they're going for, and which a lot of textbooks do, is to say that the blue chromosomes came from who? This individual's male parent. Okay, this individual's male parent, and the red chromosome then came from the female parent. Right. Okay, so we see there's 2 sets. The point is there's 2 sets, the blue set and the red set. Okay, 2 sets, that's a diploid cell. Then that means it's 6 chromosomes. Okay? All right. Now we're going to do meiosis. But before we do that, what happens? Has to happen in DNA replication. That's included in this figure as well. So chromosome replication means DNA replication. Same deal. We get replicated chromosomes, right? And now moving into meiosis, 1 and prophase 1, the chromosomes will pair. Pair. Chromosomes pair. All right? Which chromosomes pair? The homologous chromosomes pair. Right. Could you tell how this was going to happen? Maybe? I'm guessing that's a homologous pair there, the 2 big ones, right? I'm guessing maybe the 2 medium ones, homologous pair, the 2 small ones, homologous pair. That's just me, but I think that's probably what happened. And that's what happens, right? But notice that each chromosome has been replicated, absolutely, because DNA replication happens. So these are replicated chromosomes, but indeed they have pairs. Again, metaphase 1 then takes those pairs, aligns them in the middle of the cell, okay, along the metaphase plate, if you will. And anaphase 1 separates the pairs. Okay, again, are we diploid or haploid now? Haploid. We had 6 and 6. Right? 1, 2, 3, 4, 5, 6. And now 3 chromosomes per cell. We are diploid. Why do we need to continue? Because we have replicated chromosomes. Right, they were replicated. So now we need a meiosis 2. I mean, do we even need to? This is enough room right here. Separate sister chromatids. We're done. That's meiosis 2. Giving us 4 daughter cells, each of which has half as many chromosomes as the parental cell. We're done. Once again, question about meiosis. Okay, so what this figure offers us, apart from the condensed version of meiosis, focusing on meiosis 1, is also to show us mitosis. Okay, so like the case with mitosis begins, or even before mitosis happens, we do DNA replication. Again, indicated. And that gives us replicated chromosomes. Now this is now prophase, but notice, chromosomes are condensed, presumably, but not paired, right? No pairs, because this is not meiosis. In metaphase, mitosis, individual chromosomes line up in the middle of the cell. And then in anaphase, we separate sister chromatids, and then we're done. We get 2 daughter cells, each of which has half as many chromosomes as the parental, excuse me, don't think of my husband. Each of which is exactly like the parental cell, each of which has 6 chromosomes in it. Question? All right, really good figure, I think, to kind of compare and contrast the two. Maybe 15, 20 minutes, I think, just looking at that. Good figure. Okay, so we're done with meiosis. Let's close this chapter out by talking about this. Okay, what is this? Okay, siblings. I appreciate that you recognize that. Logan, I say this every semester. I probably could stand to update this figure a little bit, but guys, 15 years ago, this thing was landed. Okay, all right. So the point being, it is siblings, which you may or may not be familiar, but fictitious or not. The Obama sisters, so young. Okay, all right. So the point is, all of these individuals had the same parent, right? All of the siblings did, that is. So why are they not identical? Same parents, but not identical. Now, some cases there's some resemblance maybe going on in the family, but there's also differences, and it's never perfect. And sometimes completely different, maybe appearance or attitude, or I don't know, but clearly something is going on. So let's talk about it now. There are several things at work here. Actually, several things, namely 3 mechanisms generate variation going from one generation to the next. 3 mechanisms, variation from one generation to the next. And we'll start with this. So one thing I might ask you to do on the exam is, I said a little bit, maybe show you a diagram and ask you, what you're looking at, right? Is this, what stage of cell division, right? A little bit, not a ton of that, but a little bit. So let's do that now. Is this mitosis or meiosis going on here? Okay, I heard both, I think. Let's assume more people said meiosis, right? Okay, because indeed, the chromosomes are paired. That only happens in meiosis. So this is meiosis. Okay, if you got it wrong, you have an opportunity to redeem yourself. What stage of meiosis is it? Metaphase 1. Yes, very good. It is metaphase 1 because homologous pairs have lined up in the middle of the cell. Okay, very good. Now notice also that this is metaphase 1 in that cell. And there's the same number of chromosomes, the same species, I think. What's different? Anything? Okay, so what is different is how the paternal and maternal chromosomes have aligned along the metaphase plate, right? Whereas in one cell, both the paternal chromosomes on one side, maternal chromosomes on the other. In the other, it's different, right? It's switched. We got paternal, maternal, let's say, and maternal, paternal, right? It's reversed. Interesting. So which one is going to really happen? Which one happens?
Huh?
Okay, so I've heard some different answers there as well. The second one happens. I heard both of them happen. So this is what I like to call your gonads forgot. It's true. Your gonads forgot. And by that I mean your ovaries, your testes don't know which chromosomes came from which parent. They don't. Is that from mom or from dad? They don't know. Your body Y doesn't know that. Okay, so when they line up chromosomes in meiosis one, it's going to be random how they line up. It could be paternal on one side, maternal on the other, but it could be, you know, kind of a little above, right? And I guess it depends how many chromosome pairs you have. You can see it could be quite a complex pattern. Each chromosome lining up independently of the others. Okay, this is consequential, folks. There's two possibilities in this case. Equally possible, and it's consequential. Just consider what happens when you separate the homologous pairs and then separate sister chromosomes. Meiosis done. And what was the consequence? So daughter cells with different combinations of maternal-paternal chromosomes, right? So we have some with only maternal, or excuse me, paternal. Boys like blue, girls like red. Okay, so paternal chromosome. Some gametes have only maternal chromosomes. Some have both, a paternal and a maternal, but this has a maternal and a paternal. So in fact, there's 4 possible combinations for these two homologous pairs, isn't there? Yes, there is. All right, so this principle is known as independent sorting or independent assortment is what they say, right? And it just means that each pair lines up independently of the other pairs. I changed the wording on my slide a little bit, I wanted that on there. Each pair lines up independently of other pairs, and so it can be whatever, you know, arrangement it is. Each pair lines up independently, and you get independent assortment of the pairs. And then the outcome of that is the gametes will contain a random mix of maternal and paternal chromosomes. Okay? Question? No? Okay. So let's think about this now. How do we know how many combinations there are? And it would be more for more pairs, right? So there's a simple formula to calculate the number of possible combination of chromosomes. It says 2 to the nth power. We can use that n now. 2 to the nth power, where n is the number of chromosomes, what again? One set, right? The haploid number of chromosomes. All right, so what was n in the previous example? Let's take a look. What is the haploid number of this individual? How many chromosomes in one set? 2. 2, yeah. The maternal set has 2 chromosomes. The paternal set has 2 chromosomes. Okay? Is that clear? Haploid number is 2. All right, so it's 2 to the 2 then, right? 2 to the second power, which of course is 4. 4 combinations is what we saw here, right? 1, 2, 3, 4. Okay, very good. Now, what if there was a third pair, right? What if the number of chromosomes in a set was 3? Then what would the combinations be? Would be 8, right? 2 to the third power, yeah, is 8. Okay, now it's already doubled. You went up 1 pair, but it doubled the number of combinations possible. What is our haploid number? 23, right? And what is 2 to the 23rd power? All right, I'd be surprised if you could do that, but it is a very large number. It's 8 million. It's 8 million. Do we even need to go on talking about ways that generate variation? So what we're saying here is that an egg is 1 in 8 million possibilities. A sperm is 1 in 8 million possibilities. Of course you're going to be different from your siblings. How would you possibly have the same combination, right? And on top of that, any sperm could fertilize any ova. Any one of these 8 million would fertilize any one of the other 8 million, right? In theory, right? So this is going to be a way to generate variation, right? Any sperm can fertilize any ova. Fertilization is random. So, for example, again, for n equal to 23, as it is for us, there's 8 million possible egg, 8 million possible sperm, for how many possible combinations? Again, good luck if you can do, I mean, more power to you. I can't do that, I'll just tell you, I've done it before. It is 70 trillion possibilities. So a zygote is 1 out of 70 trillion possible combinations of chromosomes. Again, do we need to go any further? Because of course, if we're 1 out of 70 trillion possibilities, how would, having a few siblings result in the same combination? No chance, no chance. But we're not done. Oh, sorry, question, Haley? Okay, so the question is, although there are cases where you do have the same chromosomes, the case of identical twinning, right? Well, in that situation, you had a single zygote with that combination of chromosomes, which first split into 2, and then began the mitotic cell division that produced the embryo and the. So it's a very unusual case, but it doesn't really change the fact of what we've said here. Okay? So we're saying that there's a couple ways variation arises, one from independent assortment, then of course from random fertilization. There's a third way, and it's the following. Notice that in these combinations we looked at earlier, all of the chromosomes are either paternal or maternal. You might have a mix, but it's either paternal or maternal, right? Completely paternal or maternal. As opposed to what? It's often not that case. It's often not a case where you have entirely paternal, entirely maternal. Why not, I guess, is a fair question. What did we say happens in prophase 1 again? Well, what we said was that chromosomes pair, right? What we didn't say was how. I hope that bothered you. Maybe a little bit. It's like, oh yeah, chromosomes pair. Guys, I mean, how does that happen? Well, because the book shows it. Yeah, come on. This is remarkable. Again, it only happens in meiosis. And I trust there's a mechanism for that. I mean, that's what this is about, folks, is understanding everything has a mechanism at the end of the day, right? So, that said, we're not going to talk about it because it's a little bit beyond this course, right? But there is a way that the cell, so your cells have forgotten which are maternal and paternal, but what they can do is compare the chromosomes so that they match the ones that are homologous, right? And it's not just because they're big, right? They're 2 big ones, I'm going to put them together. No, it looks at the DNA and says, oh, these are a pair. Oh, these are a pair. These are a pair. So there's a process at work there. And long story short, that process has an odd mechanism, and a rather drastic mechanism, in which the chromosomes are physically broken. And by that I mean the DNA is physically broken. Not to worry, it's then rejoined. It's restored. However, it could be where you're rejoined with your homolog. Okay? So that is then called crossing over, which somebody said earlier. The chromosome is restored, but it's joined to the homolog. Okay, so what this is going to do then is create not so much a new chromosome, but a recombined chromosome, where all the same genes are there, but it's a different combination of alleles. So the same genes are still there, but a different combination of alleles. Creates chromosomes with new combinations of alleles. Simple diagram just to kind of understand this. Again, early in meiosis, in prophase 1, homologous chromosomes are paired. How does that happen? It's a mechanism that's known as DNA recombination. If you want a term for it, it's called DNA recombination. It's able to match the homologous chromosomes. And that process has the consequence of breaking the chromosomes and rejoining them, but that can happen such that you do that. Okay, so now this chromatid, right, which will then become a chromosome, right, is both paternal and maternal. This chromatid is both maternal and paternal. And whatever genes are here are still there, but the alleles have been shuffled. Does that make sense? Remember, we got different gene versions sometimes, and then that becomes consequential. Okay, so again, that's crossing over. So that concludes the mechanism. Questions? No. Okay. So what we just said was that there's 3 mechanisms generating variation, and these are crossing over, independent assortment, random fertilization. And the reason I listed them that way is this is the order in which they happen, right? Crossing over happens in prophase 1, which is when the homologs get together. Homologous chromosomes are paired. Independent assortment is about how they line up in metaphase 1, how the pairs line up, and then how they're subsequently separated in anaphase 1, right? So that's going to come, so prophase, and then this is more metaphase, anaphase, right? So that's the correct ordering, and then fertilization is not even meiosis, right? That's after meiosis. After you get an egg, after you get a sperm, fertilization happens, right? Okay, so that's the order in which they work to generate variation. Again, the first 2 occurring in meiosis, the last one being part of fertilization. Now, in full disclosure, there is a 4th mechanism that generates variation. And the 4th mechanism does not merely shuffle alleles of genes, like we saw here, but it creates new alleles. It creates whole new alleles. And over time, so you get new alleles and you get new traits, right? You get new, you had only brown eyes and suddenly you have blue eyes, right? A blue-eyed individual. And everybody else has brown eyes and what's other blue eyes, right? Just a new trait all of a sudden. And over time, this process can lead to further change and ultimately to new species, right? This is the process by which new species come about. What is this process called? Mutation, right? It's a change in the genetic code. Mutation changes the code and that creates new alleles. Okay? All right, mutation is relatively rare thing. It depends, it's kind of how you look at it. It's relatively rare. So I added to the list and made it 4, but please understand that from one generation to the next, it's 1 through 3 that are typically working, to create variation among offspring. Okay? So you are different from your brothers and sisters because of these 3, not because your brother or sister is a mutant. Okay. All right. Any questions about meiosis or variation? If not, all right, turn the page. Let's start a new topic. Let's do that now. So this topic of classical genetics is actually a revisiting of our last topic, meiosis, which we just concluded. Okay, I'll show you how that is here in a moment. Like our last topic had chromosomes in it, I put chromosomes in it. Again, I'm going to say genetics and chromosomes because ultimately it's about our chromosomes, right? Which are what make us what we are. So we have to focus on our chromosomes as we did during cell division, once again, in the course of genetics. So while we might be talking about botany, it's still about chromosomes. While we might be talking about fruit fly mating, it's still about chromosomes, okay? And in the course of this section, we will visit Mendel's work, actually beginning today. I know you know who Mendel is. Mendel, known as the father of genetics, he's called, worked out basic principles of genetics, and two in particular, law of segregation and law of independent assortment, they're called. You see where I'm going with that. Okay, so you know what this is already, but this is how, so this is about how geneticists arrive at conclusions about what meiosis is about and what chromosomes are about without even knowing about meiosis. Without even knowing about chromosomes, they're figuring out the law of segregation, the law of independent assortment, okay? Then we go on and talk about some additional principles that Mendel perhaps did not recognize in his work, but came later. Then we move on to the next chapter. At that point, look at another geneticist's work, and that's working with a different organism at that point. And then we get some other principles there. And this was where, by the way, science recognized that genes are on chromosomes. So genes give us our traits, but genes are on chromosomes, those little colored bodies that do a dance during cell division, right? That had to be recognized. That connection was only made after Mendel, and once that was made, then you can get some other, once you know where the genes are and what they are, then you get some other things like crossing over. So we'll revisit all of this from meiosis in this chapter, okay? Okay, once again, I used to who's that? Who's that? It's like, yeah, it's enough. Siblings, right? The point was that these are siblings, and while they do bear some similarities, they also bear differences. So they could in fact resemble parents and each other, but they're also going to be different from both, right? So the question is, how exactly does inheritance work? Once again, we've already talked about it, right? We finished meiosis. So you've got to pretend now that you don't know about meiosis, because that's where these folks were. That's where classical genetics was when it produced the conclusions it did. Well, how does inheritance work? So in the late 19th century, there was an idea about this that basically said that whatever we give offspring is going to blend like paint blends. Now, this is the analogy your textbook uses, and I think we should hold fast to that, right? So whatever we give offspring, obviously, you don't need to be a geneticist to know you give something to your offspring, right? Something when you give a sperm, something when you give an egg, right? And all sexually reproducing organisms do that. And whatever that is, and whatever those things contain, it blends like paint blends, okay? And I mean, it's not hard to come to this conclusion, right? Because, you look at individuals in our species, and yeah, they kind of look like this parent and that parent. I can see how it's a blend, right? I mean, there's so many examples in nature where, yeah, the offspring look like a blend of their parents, right? So why is the genetic material not blending, right? Why is the genes not blending? Well, you don't say gene, but whatever that is, right? And here's a good example, a very simple example: snapdragons. Okay, these are the flowers of the snapdragons. Okay. Those of you who know, something about botany, that's not me. But if you cross red snapdragons with white, you get what? You get pink, apparently, right? And that is exactly the kind of observation that would lead someone to think, Well, I guess the whatever that's in the gametes must be blending, right? So this is known as the blending model of inheritance. What you give offspring blends with what your partner gives the offspring, right? Okay? So is it inheritance by blending? Is this right? Blend the what's in the egg and the sperm gets blended? Is that right?
No.
Like paint blends. Stick with that analogy. No. No. Our DNA is not paint. Can we agree on that?
Yeah.
Yes, absolutely. It's not. I'm telling you now, blending is a false model. And let me double down. Blending is just wrong. There's no circumstance under which blending suddenly is right. It's wrong. It's a false model because DNA is not paint. Okay? Fair enough? Okay. So enter Gregor Mendel, who of course turned this idea on its head. I think everyone knows Mendel was an unlikely geneticist and father of geneticists, right? He was a monk, right? An Austrian monk, and this is the monastery, I think part of the building there, and I think this is like the garden where he what did he work with? Pea plants, right? That's where he did his gardening and did his studies with pea plants. So the textbook gets into more background, more interesting information about Mendel, to be sure. I think I'd just like you to know that Mendel was a botanist, to be sure. He was a mathematician. He was schooled in mechanics and in statistics and very knowledgeable about these topics. He did not, however, have knowledge of chromosomes, and he did not have knowledge of DNA. Again, interestingly, arrived at principles of genetics without any knowledge, principles of inheritance, without any knowledge of these things. Chromosomes, cells, I don't think even on the microscope. Forget about knowing about DNA. Okay, so long story short, he did conclude that the blending model was wrong, which we just said that, right? But Mendel is the one who recognized that first, and recognized it in a formal, a scholarly way, right? Doing rigorous experimentation. He presented his data in 1865. The next year, he published the data. And how was this data received by the scientific community? Anyone know? They didn't believe it, right? I really would love to learn more about this. It's been a while since I've studied Mendel and his work. I'm just curious if anybody believed it, like he had a friend or buddy or something, because nobody believed this. And he was not appreciated in his time. He expired, I guess, a couple of decades later at the ripe old age of 61. That's pretty good, right, for 19th century living. And then another 15 years after that, whoops, he was right. So interesting. How, if not appreciated in his time, is Mendel now appreciated? I mean, how do we know, in fact, that he did anything on genetics? He published it in the scientific literature. There it is. Oh, I did it first in 1900. No, you don't. Look at the paper. There it is. There's Mendel. There it is. In his pea plants. So the lesson here being, get it out there. Publish your work, even if you don't have tremendous support for it, right? And among others, right? Publish. All right. That said, again, the book gets more into Mendel and the likely geneticist he was. So why is pea plants? Let's talk about it. Pea plants were a very powerful genetic tool that Mendel used, and understand that in science, we sort of gravitate toward tools and organisms that are easy to use, right? And which can represent all the other organisms that are hard to use, right? Or which we have less knowledge or information about, right? So peas are eukaryotes, right? So they can represent other eukaryotes in that capacity. Peas are multicellular, so they have some commonalities with other multicellular organisms, namely they reproduce sexually. It's a sexually reproducing organism. So understand, it's going to represent other plants, but it's also going to represent what? Animals, humans, sure, but other animals as well that reproduce sexually, okay? Moreover, peas are cheap, they're easy to handle. The time between one generation and the next is short, so you can see the offspring more quickly and arrive at conclusions more quickly. And they have obvious characteristics with distinct varieties. Okay, this is important. Obvious characteristics with distinct varieties. Let's actually take this one over to the next slide because I want to make it very clear. Peas have obvious characteristics or characters. In genetics, you just say characters. It's just a characteristic. You know, how tall are you? You know, how many fingers do you have, right? What's your eye color? I mean, these are human characteristics, but we all have them, right? And in the case of pea plants, those characters have distinct varieties. A character is, again, just a characteristic. So if we're talking about peas, that could be flower color. Peas have flowers, and flower color is a characteristic of pea plants. A variety would be red flowers. Or white flowers is another variety of the flower color character. Okay? Another word for variety is trait or phenotype. These all kind of mean the same thing. Okay? So this is going to make pea plants a very powerful tool with all its characteristics, with the varieties. Okay? Let's look at the example. I think I pulled this from another book, but it kind of makes this point. So for example, what is a pea anyway? What is that about? What is a pea? The seed, right? Okay, the pea is a seed, and the seed in peas can have a shape that's round, or it could be wrinkled. Okay, so the seed shape character has a couple varieties, round and wrinkled. The seed can be different colors also. So the seed color character has a yellow variety, but also a green variety, as you know well. Okay, and it's not to say that you can only have two varieties, right? If you think of eye color, obviously you can have many varieties, but in pea plants, the characters that Mendel looked at all had just two varieties. I think that made it simpler. I think that really simplified his task in understanding how inheritance works. It was helpful to him, this simplicity of only having two varieties. Okay, is this terminology clear? All right. The other advantage, there's one more advantage, right? And we'll conclude with this advantage. You can make them selectively. So normally, pea plants are hermaphroditic, right? They have male and female components. They pollinate themselves, right? They fertilize themselves. Okay, so that's typical, self-pollination. But you could take the pollen from one pea plant and then, dust it onto the other pea plant. You could cross-pollinate, right? And that's what Mendel did to see what happens, right? Okay, so that's another advantage there. In his experiments, he always started with true breeding varieties. Important to understand. Everyone understand true breeding? So true breeding means if the plant self-pollinates, the offspring will always look like the plant, will look like the parent. If the plant self-pollinates, the offspring will look like the parent. And their offspring will look like what? Like the parents, right? They just never produce anything else, right? So offspring look just like the parent. If you self-pollinate, you're only ever going to see one variety
for every character. Okay? True breeding. So let's look at the typical experiment then. The idea would be if you take two distinct true breeders and cross them, what do you get? Right? So he would take a true breeder number one, a different true breeder number two, and cross-pollinate. And then what do I get? Right? Well, what you get is a hybrid. It's now something that has the genetic material of both of those parents. We call that a hybrid. Now, the experiment's not done. He would then allow the hybrids to self-pollinate. First cross-pollinate and then self-pollinate to get another generation of offspring. And then these he would analyze and try to understand what's going on. Okay, these are the ones he studied. So we got 3 generations we got to keep straight. The first generation is traditionally called P, like letter P, right? The hybrids are called F1, their offspring are called F2. If the F2 had offspring, what would they be called? F3s, you get the idea. Okay, so this is the jargon, so you know what you're talking about. Okay, shall we take a look? Yeah, let's do that now. Let's look at his first of 2 experiments, which produced one of these principles, the first one listed there. Okay, so there's only 2 experiments Mendel did that really produced his conclusions, right? So let's look at experiment number 1, and let's use the example of the P shape. The P shape. Remember, the P shape could be round or it could be wrinkled reset, right? So that's the P generation, right? Round true breeder, wrinkled true breeder. And that's a cross-pollination. And when he crossed these, he got an F1 generation of offspring. All of which has round seeds. Interesting. Self-pollinate the F1 to generate an F2, and he got, again, a lot of round. Although, wrinkle came back. Wrinkle, there it is, it came back. It was there, presumably, in the F1, and it came back. And Mendel, of course, being steeped in mathematics, also recognized, well, if I take the large number and divide it by the small number, I get 3. I get a 3-to-1 ratio, which he saw as he continued to go from character to character to character, and traits and traits and traits. He saw this again and again and again. So with the seed shape, in the P generation, you can be round or wrinkled. He got a 3-to-1 ratio, right? 3-to-1, as we just said. He went on to seed color, which could be yellow or it could be green. In his F2, he saw a lot of yellow, but also a little green. I mean, you can do the math here without a calculator. Again, it's a 3-to-1 ratio, right? And again, for flower colors, he saw, again, in each of these cases, in the F1, only one variety, and in the F2, more of one than the other, and a 3-to-1 ratio. Okay, down the line here. Okay, so clearly whatever he's seeing has some significance, right? There's some principle he's recognizing there. So what are the conclusions from experiment 1? And again, you have to pretend we haven't talked about meiosis. Is Mendel seeing the results you would expect from blending? No. Why not? What would you see in the F1 with blending if that were a true thing?
Round and wrinkled.
Something in between round and wrinkled. No, but these are all round. This is not in between. Moreover, you would not see round self-pollinating and then suddenly wrinkle comes back. How did that happen? Well, it's not blending because blending is a false model. Okay, so number one, results not expected by blending. The characters are produced by whatever is producing them, some factors that are inherited from generation to generation that are discrete. Okay, the characters are produced by factors that are discrete. Do you know the meaning of that term? It means solid. It means unchanging. It means it's not paint. It doesn't blend like paint does. Clearly does not do that. Okay, what are factors? What is he talking about? Genes. Right? Factors are genes. That's how he refers to them. But he's talking about genes, and he's saying genes produce characters, but they don't blend in offspring. Okay? Now, clearly, the F1, which is all round, still has wrinkled in it, because otherwise, how does it produce offspring that are wrinkled? So the simplest conclusion is it's round because it got a round gene from its parent here, but it also has wrinkle in it because it got a wrinkled gene from the parent there. And so it has two. Clearly it has two. So an organism has two of each gene, which produces a character. And we get one from each parent. Today, of course, we know that we inherit a pair of what? Homologous chromosomes, right? Homologous chromosomes have the same genes on them, so we get two of each. We get two of each. Now, why are there different phenotypes for each character? Well, clearly a gene has alternate versions. I don't think that was a big reach at that point, right? So let's make that a conclusion. Genes can have alternate versions, and we call those alleles of genes. We already used that term before. So for example, a round allele, right? A wrinkled allele, all right? And today we know, of course, a gene can vary in its what? Sequence of nucleotides, right? And that's what gives us different alleles, all right? So clearly there's something about round, right? Why is that all that we see in the F1? Why is it mainly in the F2? So we need terminology there at the very least. So we'll call the round seed phenotype the dominant one, right? Oh, and then the wrinkled seed phenotype the recessive one, right? It's appropriate to what we see, okay? And then likewise, there's a wrinkled allele in the F1, but it's not seen, right? So we can use these terms for the alleles as well, dominant allele and recessive allele. So if there's two different alleles together, the dominant allele affects the appearance, and the recessive allele has no effect on the appearance. Okay, now one more minute. Bringing us to conclusion 5, alleles we have two of, but separate when we go to reproduce. Separate when we make gametes. And this is known as the law of segregation, right? Which just means separation. Today, of course, we know homologous chromosomes will separate during meiosis, and this is particularly important when you have two different alleles. If you have two of the same, big mystery, what do you give your offspring? But two different ones, it's very interesting. Okay, so that brings us to the final model that Mendel arrived at, which says the genetic material is composed of units, which we call genes today, which are passed on to offspring, but which retain their identities in the offspring. They're not blended like paint blend. Okay, this is called particulate model. Particulate model of inheritance. Question. All right, folks, nail it down. We'll pick it up with this experiment and then his number 2 on Monday and go from there. Okay, we're doing that.